My youngest has like the same foot as me. I got the same weird, creepy angling of my toes and whatnot. It's creepy, but he is not exactly me. So even though a lot of stuff may be very similar from one offspring to the parents, there's still that just a little bit of variation that exists on a multitude of various different levels. All right, so let's look at the individual phases of meiosis here. We have meiosis 1 and meiosis 2. Remember, there were two phases of it, two divisions. Okay, meiosis 1 is the first. Meiosis 2 is the second. And we distinguish it by that Roman numeral right there. Okay, 1, 2. Okay, so when you're talking about the first division of prophase, you call it prophase Roman numeral 1. Metaphase 1, anaphase 1, telophase 1. And then it enters what we call cytokinesis. Okay, this is this in-between station between meiosis 1 and meiosis 2. Okay, it doesn't enter into the duplication of S phase or anything like that. Okay, there's no phase of interphase necessarily between the two divisions. Okay, it just stops and holds position. We call it rest. Then it goes through prophase 2, metaphase 2, anaphase 2, telophase 2, and then I have my end result, which is 4 haploid daughter cells. Okay. So when you're talking about mitosis, no Roman numerals. When you're talking about the meiosis, you got to have Roman numerals. Don't forget that. In the past, I've asked questions, explaining some of the differences between them. And if you put, well, prophase 1 or prophase looks like this in mitosis, but for prophase and meiosis, which one are you talking about? Prophase 1, prophase 2. Okay. And that's where I have to take out, my purple pen and slash it. And I don't like slashing. Okay. I don't like doing that. So don't forget those Roman numerals. Don't give away the easy stuff. That's easy points. The 1s and the 2s. Okay. Roman numeral 1, Roman numeral 2. And as you see from mitosis, same stages in the same order. Same phases of meiosis in the same order. Okay. How easy is that for you? I mean, this is usually science where they give you a different name for the same damn thing. Okay. They've made it easy for you here. The same phases in mitosis exist in meiosis. I just put Roman numeral 1 or Roman numeral 2 after the words. Nothing new to remember here. So let's look at prophase 1. Spindle forms. Yep. Same thing happened in mitosis, right? Nuclear envelope fragments. Nucleolus disappears. All of this happened in mitosis. Yes? This is where everyone is shaking their head. Yes. Giving me the thumbs up. Okay. But now it becomes just a tiny bit different. Thank you, Celeste. Now it becomes just a tiny bit different. Okay. Homologous chromosomes start to pair up. This is something different. And I think I have charts and stuff to have all the differences or similarities between everything a little bit further back. Okay. These homologous chromosomes pair up and they align themselves side by side, just like in that bivalent picture that we just saw where it crossed over. Okay. This forms, I think I've been using the word, but I can't remember. Bivalent. Okay. Or tetrads. Usually this is what I like to call it. Okay. Here is the bivalent. Okay. That's what we have there. Okay. The bivalent. We don't see this in mitosis, and we don't see homologous chromosomes pairing up. Okay, we don't see that. Sorry, I got something in my eye here. Okay. From prophase 1, we go to metaphase 1, and my homologous pairs are at the metaphase plate. That's different from mitosis because it's usually just chromosomes. Okay. Bivalents are aligned at the spindle independently. This has its own, this has its own spindle fibers. This has its own spindle fibers. Okay, that's all that means. But it's the homologous pairs. The bivalent is lined up at the metaphase plate. I don't say that with mitosis. From metaphase 1 to anaphase 1, okay, each bivalent separates. And, we have pictures here to show this. See, there's pictures here, okay? There's my bivalent. Each bivalent separates, okay? Oh, I guess I should put here. Crossing. Sorry. Crossing over can occur here. Doesn't have to, but it usually does. Can occur here. Okay. This is where that crossing over occurs. That independent assortment, that happens in metaphase 1. Okay. Independent assortment. Independent assort. A-S-S-O-R-T. I'm not going to spell the whole thing. Okay. Bivalents separate. They go to each pole, just, similar to what happens in anaphase, right, of mitosis, but now it's the bivalents are separating, okay? Sister chromatids do not. Sister chromatids do not separate because that's what it does in mitosis, right? Okay, so I still got my chromosome, okay? Still got my chromosome. Daughter cells, they kind of separate, they'll pinch off and, cleavage furrow and ta-ta. And then it enters into this thing called interkinesis. As you can kind of see here, I didn't put nucleolus comes back, nuclear membrane reforms, chromosomes decondense or uncondense into chromatids. Okay. I'm sorry, chromatid. I don't see any of that. The chromosomes separate and they wait. If this is a dude and we're talking about sperm production, those two suckers will just start going into the next phase. Okay, prophase 2. If this is a woman and they've made it this far, they arrest in a different phase. They stop and then they'll finish and then they'll stop and then they'll continue and then they'll stop. And women gametogenesis is just much more complex than a guy. So we get into this thing called interkinesis where we just enter this holding pattern, okay? How long that holding pattern is just depends on if you're a woman, if you're a man, okay? There's the crossing over, there's the independent assortment, there's the bivalents, there's the bivalent separating, and I've gone doon, 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 doon. Okay, I separated out the chromosomes. Okay. Then these dudes are going to enter into meiosis 2. Everyone still with me? Cool. All right.
Meiosis begins after interphase, in
Let's turn this up a little bit.
The chromosomes of a
Let me turn this up a smidge, guys. I apologize.
Meiosis begins after interphase, in which the chromosomes of a diploid cell have replicated during the S phase. After the 2 successive cell divisions of meiosis, a single cell will produce 4 cells, each having a haploid number of chromosomes. Prophase 1 of meiosis 1 begins with the condensation of chromosomes and the fragmentation of the nuclear envelope. Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, homologous chromosomes align next to each other to form tetrads. A tetrad is composed of 4 sister chromatids. Crossing over will occasionally occur between the non-sister chromatids in a tetrad. During prometaphase 1, the spindle apparatus continues to form, and the spindle fibers attach to proteins at the centromeres of the tetrad. One pair of sister chromatids is connected by a spindle fiber to one pole, while the homologous pair of sister chromatids is connected to the opposite pole. The chromosomes align at the metaphase plate during metaphase 1. Notice that the tetrads are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase 1 involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the spindle fibers shorten. When the sister chromatids reach the poles of the cell, they detach from the spindle apparatus and the nuclear membrane begins to reform.
Not in every cell.
A cleavage furrow forms, and cytokinesis results in 2 cells, each having a haploid number of chromosomes, with each chromosome consisting of 2 sister chromatids. This completes meiosis 1, and after a period of interkinesis, the 2 daughter cells start the second cell division cycle called meiosis 2.
All right. It's similar, but is shorter and it's a phase of quote unquote rest. And in terms of being similar to interphase, that's about it. Okay. I mean, it says it's similar, except it's shorter. Okay. Yeah. And DNA replication doesn't occur. Okay. Yeah. Okay. So, I mean, is it all that similar? I don't know. Whatever. Okay. It's just a brief interlude, if you will, between meiosis 1 and meiosis 2. Chromosomes will condense back again in some cells. In some, it goes immediately from telophase 1 into prophase 2. Okay. So if you want to say it's chromosomes condense, okay, then yes. Right? In those couple examples, it is exactly the same. Chromosomes condense. What else happened in prophase of mitosis? Nuclear membrane fragments. Nucleolus disappears. Okay, so prophase 2 is the exact dang thing that happens in mitosis. Metaphase 2, chromosomes align at the metaphase plate. Okay, not homologous pairs. Chromosomes align at the metaphase plate. Same dang thing as mitosis. Anaphase 2, sister chromatids separate. Remember that centromere that holds the 2 sisters together? Dissolves. Sister chromatids separate. Just like anaphase of mitosis. Telophase 2. Nucleolus comes back, nuclear membrane, yada, 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 uncondensed down. There's my 4 haploid daughter cells. If they've crossed over, they are all very genetically unique. Okay. They've independent assorted. They are genetically unique. Okay. And that's what this picture here kind of shows. Okay. Yep, chromosomes. Chromosomes line at the metaphase plate, not the bivalent, not the homologous chromosomes. The sisters separate at their centromere. One goes to each pole. There you go. Cleavage furrow. Doubling up. 4 haploid daughter cells. Yeah. Meiosis 2 looks exactly like mitosis. So if you know mitosis, you know meiosis 2. Okay. So if you know mitosis, you basically know meiosis part 2. This is just showing you what they look like. These are 2 individual cells, well, 1 cell becoming 2. And then 2 cells dividing in half again. And there you got all your haploid daughter cells. Pretty simple, right? Of course. Of course.
During prophase 2, the chromosomes condense again, and the nuclear envelope fragments. During early metaphase 2, the spindle apparatus forms, and the sister chromatids become attached to the spindle fibers. In this picture of early metaphase, a pair of sister chromatids, a dyad, is attached to both poles, not just one pole as in metaphase 1. At metaphase 2, the dyads are aligned along the spindle equator, with each chromatid attached to one pole. The sister chromatids of each dyad separate and move toward the poles during anaphase 2. During telophase 2, the nuclear membrane reforms and chromosomes decondense. In animal cells, a cleavage furrow begins to form, leading to cytokinesis. With the completion of cytokinesis, meiosis II results in the formation of 4 haploid cells. Thus, meiosis begins with a diploid parent cell that has replicated its chromosomes and ends with 4 haploid cells containing 1 unique set of chromosomes each.
So like I said, okay, a good potential short answer question is, describe the phases of mitosis in the correct order. Keyword being describe, not list. Another good question is comparing meiosis and mitosis. Okay, well, let's look at that. Meiosis, 2 divisions. Mitosis, only 1. In meiosis, I can get synapsing and crossing over. Okay, chromosomes here do not synapse or cross over. Okay, centromeres dissolve during anaphase. That's what allows those chromatids to separate. Well, they can survive here. See, centromeres survive in anaphase 1. That's why it's so important to remember those Roman numerals 1 and 2. Because if you put nothing there, you'd be wrong. If you put 2 there, you'd be wrong. Okay, so you got to have those 1s and 2s. Don't give away easy points. We half the chromosome number. Well, in this one, we preserve the chromosome number. They are identical, right? 4 daughter cells, 2 daughter cells. Genetically different. Awesome. Genetically identical. That's awesome too, if I want it to be skin, right? This is sexual, and this is asexual. Okay. Wouldn't it be a fantastic question if I asked you to explain or compare and contrast meiosis and mitosis, or mitosis and meiosis? Okay. Of course it would. Because if you could talk about it, comparing them, then that tells me you know both of them. And I don't have to ask what are the phases of mitosis in the correct order from beginning to end to tell me one or two things that occurs in each one. I don't have to do that. If I ask you just to describe what's going on or what's the difference between or what's the similarities and differences compared with meiosis and mitosis, see, there's a lot of ways to ask it, right? A lot of ways to ask it. Then hopefully you'll remember slide 30. These are some similarities, okay? These are some similarities here, all right? Because that previous one, I guess, looked at differences. So if we looked at similarities, I mean, they both involve an orderly stage of events: prophase, metaphase, anaphase, telophase. It includes them all. They all go from one to the next. And there's a series of things that happen in each one. So from that standpoint, yeah, similar. Spindle fibers were there, similar. They do the same thing. It may be independent assortment, but they're assorting. Cytokinesis is involved with some, not with all, but for all intents and purposes here, there's cytokinesis with each. Okay, there's cytokinesis with each.
DNA replication occurs prior to both meiosis and mitosis. In both processes, the nuclear membrane breaks down as the DNA condenses into chromosomes. In meiosis, homologous pairs of chromosomes, called bivalents, come together during synapsis. Crossing over can occur, which results in a mixing of the genetic information between the chromosome pairs. In meiosis, the bivalents then align along the central plate of the cell and subsequently separate, one chromosome traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involved a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
Here is a great chart that shows you a comparison, between one and mitosis. Pairing of homologous, no pairing of chromosomes. And this is kind of a synopsis of what I was talking about when I was going over these steps. Okay. Pairing of homologous. Sorry. No pairing, okay, of homologous here. We have bivalence at the metaphase plate. It's just a duplicated chromosomes. There's no bivalence associated with mitosis. Okay. Bivalents separate. Okay. Sister chromatids do that in mitosis. 2 haploid daughter cells, not identical. 2 diploid daughter cells that are identical. Well, what if we looked at mitosis 2? Okay. Compare that with mitosis. Okay. Yeah. I mean, the only difference is, does everybody see the one difference? I mean, there's the one little difference, but it is exactly the same. Like I said, and hell, they didn't even talk about, synapsing or crossing over. Crossing over, independent assortment, INDEP, ASS, ORT, independent assortment. Okay. The only difference between this is, again, haploid and diploid. Okay. And that's just because, well, we're in the second phase of meiosis. I've already kind of halved the material. So we just have half of the numbers there. Okay. But I mean, it's still the same general stuff though, right? I mean, the haploid number is, but it's still duplicate. It's still the chromosomes, right? The chromosomes are at the metaphase plate. The chromosomes are at the metaphase plate. So I mean, that's the same. Sister chromatids separate. Sister chromatids separate. Okay. I don't like their wording here, but there are 4. Okay. Haploid daughter cells. They're just looking at an individual cell and not looking at the totality of it. So I apologize for their bad words. Okay. 4 haploid daughter cells, not genetically identical. Telophase. 2 diploids, genetically identical. Okay. So, maybe better questions would be, what is the similarities and differences between mitosis and meiosis? Compare meiosis 2 with mitosis, or compare meiosis 1 with mitosis. That may be a better short answer question. Okay. Asking you to compare and contrast, I mean, that's probably a little bit robust of a question. That's probably a little bit better of a question. Okay. And there's that kind of visual there that shows that. Okay. So when you're going through this, guys, go through and read the slides. Sure. And, read it over. Okay. Read it over. Give yourself a half hour. Review these mitosis and meiosis. Okay. Review the steps of it. All right. Look at mitosis and then look at meiosis and then look at mitosis and then look at meiosis. Okay. And then after about maybe 10 minutes of looking at, comparing and contrasting those two, looking at them, go look at these pictures here. Okay. Now, and just cover up the words that are there. I'm sorry, I didn't include pictures with no words. But just look at the picture and just talk out loud. Mitosis, prophase, nuclear membrane fragments, nucleol. Go through it, say it out loud. Do it a few different times, saying it out loud. Okay. By doing that, looking at it, okay, saying it out loud, looking at a picture, saying it out loud in the air, you're going to create better connections in your memories there, and you're going to be able to remember this stuff on game day, or remember this stuff on the cumulative final, right? Because there's going to be a few cumulative questions on that sucker. All right. And obviously mitosis and meiosis would be a big thing that I would ask. Okay. But do those things over and over. Give yourself an hour a day or a half hour a day, whatever you can. Just give yourself that time. Put it away. Come back the next day. Give yourself an hour or a half hour. Hell, I'll take a half hour from you. Okay. Put it away. Come back and look at it again the next day. Just keep building on it every single day. Maybe one day you looked at mitosis. The next, let's say let's just say you're giving yourself 2 hours a day. You're giving yourself a half hour to look at mitosis, giving yourself a half hour to look at meiosis. Go away. And say you had another hour later on in the day. I have a half hour. I'm going to look at some similarities between them or differences, compare and contrast. I have another half hour. I'm going to look at something else, another aspect associated with this. Okay? Some of the minutia within the note set. Okay? Not necessarily looking at mitosis and meiosis. Put it away. Come back again the next day. Okay? You're going to find that you're going to remember this crap a hell of a lot easier if you do that. I promise. I promise you. And then you don't have to ever see me again. Unless you come to nutrition class. Of course you will, because I am the best teacher. No, I'm just joking. I'm just trying to lighten the load here. Do you guys have any questions about what we did today?